Now, let us look into another type of macromolecule, biomacromolecule, that is we are having is the nucleic acids. We have discussed about nucleic acids earlier in the first class itself, where we have considered the DNA and RNA. Okay, so the examples for the nucleic acids we are having is the DNA and RNA are the examples for the nucleic acids, right? And generally we say that nucleic acids are polynucleotides. Now again the same term, polynucleotides. Okay, it means that linking of one nucleotide, okay, nucleotide with the another okay, tide, and similarly another nucleotide, and so on, right? So this will result in the formation of the nucleic acid. Okay, and now if you consider the component of a nucleotide, we have discussed this again. Okay, we have discussed in the earlier classes that a nucleotide is mainly made up of the nitrogenous base, and it is also made up of a sugar, right? Generally the sugar is actually pentose. Okay, it is mainly a pentose. Okay, an example for a pentose sugar that is mainly present in the nucleic acid is the ribose. Yes, so we actually find two types of ribose, the normal ribose, and one more we are having is the exactly deoxyribose. Yes, ribose and the deoxyribose. We are having these two different type of ribose molecules. Okay, so if you look into the difference between the ribose and deoxyribose, this is the structure of your ribose molecule. This is the, what we can say, first carbon, and this is the second carbon, right? So the second carbon, if it is having hydroxyl group, then it is called as the ribose. If the oxygen is missing and simply hydrogen is present, it is called as the deoxyribose. Okay, so the deoxyribose is mainly present in the case of DNA. It is the sugar that is present in the case of DNA, and ribose is mainly present in the case of RNA, right? Yeah, and finally the last component we see is the phosphate molecule. Okay, yes, and phosphate is the reason that is giving the DNA or RNA the negative charge. Remember, okay, DNA molecule is actually negatively charged. Okay, and the reason why DNA is actually negatively charged, because of the we can say about the phosphate molecule. It is mainly the phosphate molecule that is negatively charged, contributing the negative charge property to the DNA or the RNA. Okay, hence we can say that the backbone of the nucleic acid, be it the RNA or be it a DNA, is the phosphate backbone. But in your textbook they have mentioned that the backbone, that is the backbone in the case of the RNA or even the DNA, they have mentioned that it is the phosphate. Okay, phosphate sugar backbone. Okay, they are mentioned as the phosphate sugar backbone, right? But in textbooks, some high level biology textbooks, they say that it is just phosphate backbone. Okay, and also remember, phosphate provides or contributes the negative charged property in the case of the RNA or DNA. Okay, so now let us look into the nitrogenous bases. Okay, nitrogenous bases. So here we are having four different nitrogenous bases, right? So first one we are having is the adenine, and the next one we are having is the guanine, cytosine, and also thymine. And at the same time you also find one more base. It is called as the uracil. It is called as what? Uracil. Okay, so now cytosine, thymine, and also the uracil, they are also called as the pyrimidines. Okay, they are called as the pyrimidines, but adenine and guanine, they are mainly called as the purines. It is mainly called as the purines. They are called as the purines, right? I hope the concept of proteins and the structure of proteins and also the type of nucleic acids is also quite clear for you people. Now we can move on to the next concept, that is nature of bond linking monomers in a polymer. Okay, so we will mainly discuss the different type of bonds. Okay, different types of bonds in the case of a polymer, in a polymer, different type of bonds in a polymer, right? So we are not going to discuss the property of these bonds, okay, more in detail, but we will mainly name some important bonds that are mainly found in the biomolecules, okay, especially the macromolecules, okay. So here you find mainly the first type of bond, that is called as the peptide bond. It is called as the peptide bond, okay. Now peptide bond is mainly involved in the linking of amino acids. The property of the peptide bond is that it is mainly involved in linking of amino acids. So just a moment ago I had written that one amino acid is linked with another amino acid. So this line what I have drawn over here is actually the peptide bond. It is actually the peptide bond that we have mentioned over here. And interestingly, the result of a formation of a peptide bond, okay, so the result of a formation of the peptide bond is the formation of water, okay. Water is actually a byproduct in the peptide bond formation, okay. Now why water is actually formed? Because the peptide bond formation mainly involves two important groups, that is mainly the carboxyl group and one more group, that is the amino group, okay, amino group, right? So now one hydrogen from the amino group and OH molecule from the carboxyl group, you can see that both of them are actually released, and this results in the formation of, or release in the water molecule, okay. So hence the peptide bond formation results in the release of water. They result in the release of water, and hence any reaction that results in the formation of a bond and causes the elimination of water from the reaction during the bond formation is called as dehydration reactions. They are called as the dehydration reaction because there is release of water molecules, right? So peptide bond is mainly involved in the formation of linking the amino acids, okay. So when you start to link one amino acid with the another amino acid, this results in the formation of a protein. Yes. So the product, as we mentioned, it is mainly the water over here, okay. And such kind of reactions, it is called as the dehydration reaction, that is release of water molecules. Now moving on, we are having one more type of bond. It is called as the glycosidic bond. It is called as the glycosidic bond, and a glycosidic bond is mainly involved in not lining, it is mainly linking of monosaccharides. Exactly, sorry, it is a monosaccharide linkage, okay, monosaccharide linkage, okay. So let us consider the example of cellulose, okay, from our previous class. And cellulose, we had said that I will take one glucose molecule and I will attach it with another glucose molecule. I will take another glucose molecule and so on, right? Yes. So here you see that the line what I have represented over here is actually the glycosidic linkage or glycosidic bond. It is mainly the glycosidic bond, right? So now in the last class we had said that cellulose and starch and even the glycogen, right? Yes, all of these are actually homopolymers. It means that all of them are actually made up of similar repeating units of monosaccharide, and in all the three polysaccharide, that is cellulose, starch and glycogen, you see that the sugar that is used is the glucose molecule, right? But why we are having three different names, right? It is mainly because of the bond formation nature. It is mainly because of the bond formation nature, right? So in the case of cellulose, the bond that is formed between one glucose and another glucose is beta 1,1,4 glycosidic bond, okay. Now what do you mean by beta 1,4 glycosidic bond? It means that the first carbon of the glucose is linked with the fourth carbon of the adjacent glucose, right? Hence it is called as the beta 1,4. In the case of starch, it is actually alpha 1,4. Okay, let me write it more clearly. The bond in the case of starch is alpha 1,4. Okay, and in the case of glycogen, you see that it is alpha 1,4 and alpha 1,6, right? So yeah, so it means that there's also one more type of bond formation. It is between the first carbon of one glucose with the adjacent sixth carbon of the another glucose molecule. Okay, so it is mainly because of the difference in the linkage between the adjacent glucose molecules you see formation of different type of polysaccharide, that is the cellulose, starch and the glycogen, right? So this is an important point that you people have to remember. And now we are having the last bond is called as the phosphodiester bond, right? Now in the case of phosphodiester bond, you see that it is a bond that is linked between adjacent nucleotides. Okay, and that is mainly because of the phosphodiester bond, right? So if we have to discuss it in a proper way, the 3' carbon sugar, okay, it is linked with the adjacent fifth carbon of the another sugar molecules. Okay, so the in one nucleotide, the third carbon is involved in the bond formation with the other nucleotide sugars, which is mainly with the fifth carbon, right? That is mainly the phosphodiester bond. Now the next one we are having is the DNA structure, right? So we will study mainly about a few points about the DNA structure, right? And in the case of DNA structure, you can see that it is mainly a double helix structure. Okay, especially it is a right-handed coiled helix. Okay, so if you have to draw the diagram, this is how you draw, right? Yes. So this is the DNA molecule common diagram that all of us have studied to draw, right? And the backbone of the DNA structure is mainly made up of sugar and phosphate, and it is mainly made up of nitrogenous bases. Okay, the distance between the two nucleotides, that is this one, right? It is around 3.4 angstroms. And finally, the last one we are having is the one full turn from here to here. You see that it is mainly made up of 10 base pairs, right? And one pitch, that is from here to here. Okay, this is called as one pitch, and this one pitch is around 34 angstroms. Okay, so this is mainly about some of the salient features of the DNA structure. So now we can look into some few questions, and we can start our discussion on that. The proteins are made up of the nucleotides, polypeptides, amino acids and sugars. So we all know it is mainly made up of the amino acids. Okay, it is mainly made up of the amino acids. The next one, the protein that is present in the intracellular ground substance is collagen receptor antibody insulin. It is mainly the collagen molecule that you mainly find present as the intracellular ground substance. The next one, the hormones mainly exert their action by binding to the insulin collagen antibody receptors. Okay, we know that the hormones mainly bind to the receptors and they can exert their actually actions, right? So we can say that D is our answer for this particular question. So the next question, the amino terminal of a protein is referred as a C terminus, N terminus, reducing end, non-reducing end. So we know that the amino terminal or one end of the protein that is having a free amino group, it is called as the N terminus. So B is our right answer over here. The next question, the tertiary structure of proteins is achieved by dash bonds, okay, and that is the peptide bond, covalent bond, disulfide bond, ionic bonds. We know that it is mainly the disulfide bond, right? So one protein will be bound with another peptide or a protein, okay, and this bond we call it as the disulfide bond, right? So ultimately the tertiary structure of the proteins is achieved by not only with the help of disulfide bonds but also with the help of looping and folding of the protein, okay. That is what we had studied. The next one, which of the following is a carbon compound formed of heterocyclic rings? Okay, it is the proteins, amino acid, lipids, and nitrogenous bases. We know the heterocyclic compounds are mainly the nitrogenous bases, which are mainly found in the nucleotides. They are mainly found in the nucleotides, right? Yes, that is mainly our DNA or RNA. The next one, select the incorrect statement, and that is ribozymes have the catalytic power. It is actually a true statement, okay. Though they are actually RNA molecules, they do have the catalytic power. Okay, the next one is the proteins are homopolymer of amino acids. No, they are actually the heteropolymer of amino acids. So hence we can say that B statement itself is the incorrect statement and the right answer for us. Okay, so let us look into the two remaining options. Inulin is a polymer of fructose. Yes, it is actually true. And glycogen is stored in a liver and muscles. Exactly, glycogen is the reserve food material or stored as food material in the case of animals, right? That is liver and muscles. The glucose is stored in the form of glycogen, right? Hence the D is also a correct statement, and the wrong statement is actually B statement. The next one, which of the following statement is true? The essential amino acids cannot be produced by the body, okay. There are 15 amino acids. Non-essential amino acids are obtained through diet only, and the collagen is the most abundant protein in the biosphere. We know that it is actually the Rubisco enzyme that is the most abundant protein in the biosphere. Hence, this is incorrect. Okay, and the non-essential amino acids are obtained through the diet, as they say. It is actually the essential amino acids that we get through the diet. Okay, hence this statement, C statement, is also wrong. And there are 15 amino acids, and we know that there are actually 20 amino acids. Hence this statement is also wrong. And the first statement, as they say, essential amino acids cannot be produced by the body. Exactly, hence we get them through the diet or the nutrition what we consume. Hence A is our right answer for this particular question. Now moving on, which of the following protein is a protein and also an enzyme? So they're asking which of the following is an enzyme and also a protein, right? So collagen is a protein but not an enzyme. Ribulose bisphosphate carboxylase and oxygenase, yes, it is a protein and also it is an enzyme, right? And the next one we are having is the antibody, and antibody is just an immunoglobulin, only a protein. It is involved in the defense mechanism. It does not have any kind of the enzymatic property. And finally we are having receptors, and receptors are again only proteinaceous in nature. They do not have any kind of enzymatic property. Okay, though there are some exceptions to this rule, but generally we say that they are mainly proteins. Okay, so we can say that B is our right answer, that is ribulose bisphosphate carboxylase oxygenase. The next one, facilitated transport of molecules is done by carrier proteins that are present in the cytoplasm of the cells, vesicles, cell membrane, and lysosomes. Okay, so one example we studied in the protein, that was mainly the GLUT4, right? Now GLUT4, we all know it is mainly present in the cell membrane. Okay, it is mainly present in the cell membrane, and the function of the GLUT4 is mainly what? Enable the transport of glucose into cells, into the cells. That is what these GLUT4 is doing, and GLUT4 is also an example for the carrier proteins, and hence we can say that they are mainly present in the cell membrane. C is our right answer for this question. Right, now moving on, identify the level of the protein structure. Okay, so if you look into the diagram that has been given in your textbooks, okay, so you can see the looping as had just drawn a rough schematic representation while in the class. Okay, the looping and folding is actually the tertiary structure. Okay, this is actually the tertiary structure. Okay, so primary no, secondary no, tertiary yes, exactly, this is actually a tertiary structure. Quaternary no, it is not a quaternary structure. Next, which of the following is true? The primary structure of a protein involving the alpha helix. Okay, now the next statement is only some part of the protein is arranged in the form of a helix. Assemblage of more than one polypeptide to form a functional unit is the quaternary structure of protein, and a left-handed helix is observed in the proteins, right? So we know that it is mainly right-handed structures. Yes, it is mainly right-handed structure you mainly find in the case of proteins. Hence, this statement is wrong. Assemblage of proteins more than one polypeptide to form a functional unit is the quaternary structure of proteins. Exactly, even this statement is true. And so only some part of the protein is arranged in the form of alpha helix. Exactly, even this statement is also true. So again, you can see that two statements we have got it true over here. We can say that same repetition has occurred from our previous class. Okay. So again, we can consider this question as invalid. Okay, this question would have been valid only when we had said that all the part of the protein could form the helix. Okay, then the B statement would have been wrong, and the C statement would have been the true statement. Okay, and similarly, assemblage of more than one polypeptide to form any more functional unit is called as a quaternary. Instead of quaternary, we could have said it as a tertiary or something like that. Avaga, we can say that the C statement is wrong, and the remaining statement, that is the B statement, is true. Okay, and A statement, we know that primary structure is actually not alpha helix, it is the sequence of amino acids present in the protein. Okay, so let us consider this as invalid question. This question is invalid. Okay, it would have been valid only among the B and C options if one of the options could have been stated true. Okay, so more than two options here are true. So let us consider it as an invalid over here. The transport of glucose is enabled into the cell through the trypsin, GLUT4, insulin, collagen. Okay, it is the GLUT4. Through the GLUT4, what happens? The glucose molecule enters into the cell. The next one, the presence of sugar, phosphoric acid, and heterocyclic compound is a nucleotide. Okay, nucleoside, nitrogenous base, all of the above. It is mainly the nucleotide. Okay, it is the nucleotide that is mainly formed by the sugar, phosphoric acid, and the heterocyclic compounds. The type of sugar that is mainly present in the RNA is a deoxyribose, hexose, glucose, ribose. Yes, the answer over here is the ribose is the right answer over here. Okay, ribose is mainly formed by it is the main sugar that you find in the case of RNA molecules. The next one, the number of subunits that is present in the hemoglobin is 4, 2, 3, and 1. You see that it is mainly made up of what we can say 4 subunits. Okay, 2 alpha and 2 beta subunits. Okay, completely 4 subunits form the result in the formation of the hemoglobin. The next question, individual monosaccharides are linked by glycosidic bonds, peptide bonds, disulfide bonds, and the next one we are having is the phosphodiester bond. Okay, so here you can see that they have mainly asked about the monosaccharides. So hence the glycosidic bonds are involved in linking of the monosaccharides to give us polysaccharides. Right? Yeah. The next question, which of the following statement is not true? The peptide bonds are formed between the carboxyl group and the amino group. The glycosidic bonds are formed between the monosaccharides, okay, and the phosphodiester bond is formed between the third carbon of a sugar and a fourth carbon of another nucleotide sugar, right? We had said that it is mainly between the third carbon and the fifth carbon, not the fourth carbon. Hence, we can say that the C statement is actually false statement, and that is our right answer. The next one we are having is the glycosidic bond is formed from the dehydration, right? Exactly. The product is that the water is released. Hence, we can say that even this statement is true. The right answer over here is the C statement, which is not true over here. Now, moving on, the product that are released from by forming a peptide bond is water, carbon dioxide, hydrogen, and molecular oxygen. We know it is a dehydration reaction. Hence, water is the product that is released by the formation of peptide bond. Right? So A is our right answer over here. The next question, which of the following statement about the DNA is true? Okay, it is a double helix. The nitrogenous bases are projected parallel to the sugar and are inside. The binding of adenine is with the cytosine and guanine with the thymine, and the distance between the two nucleotide is 34 angstrom. No, the distance between the two nucleotide is around 3.4 angstrom, and the adenine always binds with the thymine and cytosine binds with the guanine. Hence, we can say that this statement is not true. Even the D is not true. The nitrogenous bases are projected parallel. No, they are actually projected perpendicular. Okay, you can look over here. Probably I may not have discussed this point. Okay, the nitrogenous bases, when they are attached to the sugar, they are projected perpendicular and they are buried inside the double helix. Okay, and the first statement is that it is a double helix. So A is our right answer for this question. So we have mainly discussed around 20 questions for this particular class, and you people have to solve more questions so that you can be more proficient in this particular section. Don't forget to discuss or to mainly emphasize on the protein section. Okay, lot of questions you people can expect in your examination, mainly from the protein section only. Okay, it's a major concept, so don't even think of skipping that particular part. Okay, so I guess we will stop our discussion over here, and in the next class we can start discussing on the remaining part of the biomolecules. Thank you.